So leaves can adapt to many different types of environments, and some of the environmental factors that can exert selective pressure on leaf traits I've listed here, such as temperature, humidity, elevation, herbivory defense, and the availability of light and nutrients, and my focus, which is the availability of water. So as I'm sure you know, plants can, variation can occur either in the form of phenotypic plasticity or as a heritable adaptation across generations, which is what I'm interested in. So here I'm just showing you a gradient from humid to dry, and we can make some predictions about how leaves might look based on the habitats in which they're found. So small leaf size is traditionally considered an adaptation for a dry climate because smaller leaves have a reduced boundary layer or layer of still air around the leaf, which helps with more effective leaf cooling. And so that means in areas where light is not a limiting factor, plants with smaller leaves might survive better in drier soils. And it's been found that across diverse species that as precipitation increases, leaf size also increases. And along with leaf size, we often find a pattern with major vein density. So the veins are often closer together or more dense in smaller leaves. So if one conduit breaks, there are still others that can maintain function, so it reduces the hydraulic vulnerability. And finally, we might use this trait Turgot loss point as a predictor of drought tolerance. So Turgot loss point is just the water potential at which the plant wilts, and it's determined by the saltiness of plant cells. So plants with a more negative Turgot loss point are more drought tolerant. So, and that's also been found across species that plants with more negative Turgot loss point can maintain somatal conductance and thus photosynthetic function and growth even in dry soils. So clearly, previous studies have established in theory for how anatomy of venation traits can contribute to drought tolerance, but few studies have looked for how these traits are related to climate within a lineage, and in particular, no studies to my knowledge have looked at how traits diversified across an aridity gradient within a lineage. So those kinds of studies are necessary to determine if the patterns, the micro-scale patterns we see like within a lineage mirror what we see across diverse species. And as I'm sure you've all heard, drought has become a particular problem in California. So this is a NASA image from an NBC article where red is showing greater accumulated water loss. So plants in California now not only have to deal with these seasonal drought fluctuations that are typical in the Mediterranean climate, where there's just a hot, dry summer and a mild winter, but they also have to survive in a perpetually hotter and drier environment. So my study organism is Ceanothus, which is a genus of, it's called the California lilac because it has these beautiful purple or sometimes white flowers, but it's actually not a lilac. It's in the Rhamnaceae family or the buckthorn family of woody, usually evergreen shrubs. And it's one of the dominant genera in the California chaparral, which is the ecosystem. It's a shrubland in California. And Ceanothus also encompasses some of the most drought-tolerant species in the chaparral. And within Ceanothus, there are two subgenera. One of them is called Cerastes, and the other is also called Ceanothus. So the naming here I know is confusing. There's Ceanothus, the genus, and there's also Ceanothus, the subgenus. So I'll try to just be clear if I mean the genus or the subgenus. But the Cerastes subgenus tends to be more restricted to arid climates, whereas species in the Ceanothus genus are more widespread across North America and into Central America. So some of my questions are, which traits have helped Ceanothus survive and dominate in the Mediterranean as the Mediterranean climate emerged? And I would predict that species that are found in more arid climates would be better able to survive as water became more scarce than species that might be more widespread. And also how do leaf traits shift with habitat, and particularly with aridity within a lineage. And I predict that natural distributions of species would follow an aridity gradient according to plant traits. So I should say at this point I have a lot of data and I have some ideas. So I'm going to present how I collected the data and some preliminary findings. So sampling for this experiment took place at the Rancho Santa Ana Botanic Garden, which is in Claremont and grows only native California species. And we sampled 3 leaves per plant and mature plant and 3 plants per species for 10 species, 6 of which were in the Ceanothus subgenus and 4 were in the Cerastes subgenus. And it's just important to note that they were all sampled in this common garden, so any differences we saw between leaf traits between species we could attribute to genetic differences and not to the leaf traits responding to the environment. And we measured 56 leaf traits, and this is just an example of some of the diversity. The traits were related to anatomy, structure, and drought tolerance. And you can see that the leaves are diverse in their size, and their shape, and in their venation patterns. And we also obtained species range data from online databases and climate data from also from online databases for 12 climate factors. And I also measured the anatomy of the species, so I basically had leaves embedded in a resin capsule and then just cut the capsule to be used on the microtome, which is the machine you see here, and just like slice off a thin section from the leaf, and can view the cells and the tissues within the leaf. And finally, I used the rate calibration method to determine the divergence time between the 2 subgenera, based on the ITS region of a known mutation rate of the ITS region of nuclear ribosomal DNA. And this was done following previously published Ceanothus phylogeny. So there are 78 taxa, and we just subset 10 from that full tree. So we looked for, like I said, I have a lot of data, and we looked for 2 different types of results. We looked for variation within a trait between species, and we looked for correlations between traits and climate. And what we found was there was a lot of variation in leaf mass per area, which is what you see here, and the error bars are standard error, and the Cerastes species, which are shown in blue, tend to have a higher leaf mass per area than the Ceanothus species. And the lines here represent the subgenus averages. So this kind of supports what I hypothesized, that species found in drier places might have smaller and thicker leaves, and the Cerastes species do tend to be found in more arid climates. But it's also kind of interesting to note that there's a strong morphological divergence between the 2 subgenera. And this was mirrored in the anatomy of the 2 subgenera. So we found that the Cerastes species, which are here, tend to, rather than have their stomata on the surface of the leaf, they have these infoldings in the leaf margin, or stomatal crypts, which are basically like hair-filled pockets. So the stomata are inside these crypts. And the function of crypts is not really well understood. It was thought they could be an adaptation for aridity by keeping like a kind of moist pocket for stomata, so to reduce transpiration, and there are other ideas, but it's not really clear. But it's kind of interesting to see this, and we need to look into it more, but maybe this is just a different way that these plants are adapting to drought. And we also found that the veins were farther apart in the Cerastes species, which is not what we usually expect, but it could be just because they can only exist between the crypts, so there's veins here and here, so they might just be forced apart by this anatomy. So there's more we need to look into here. And finally, we look for correlations between traits and climate. And this is just one example, but we found correlations with several traits in climate, and here I'm showing a correlation between the Turgot loss point, or which I said at the beginning is the water potential at wilting, and precipitation. And plants with a more negative Turgot loss point were found in areas with lower precipitation, and we also found a correlation with Turgot loss point and aridity, which I haven't shown here. But this, again, kind of supports what we would expect and what has been found across diverse species. So it's exciting to also see this trend within a lineage. And we also found correlations with other traits, like I said, with like cuticle thickness and precipitation, aridity, and temperature, and leaf area and temperature. And all of these seem to hold up after conducting independent contrast analysis. So, in conclusion, we found pronounced variation in form and anatomy among the Ceanothus species and between the 2 subgenera, and I think this is maybe the first study to look in depth at the anatomy in this lineage. We also found that the leaf drought tolerance traits diversify consistently with species climatic distributions, as I showed with that example of Turgot loss point being correlated with climate. And in the future, I'd like to look at how these traits have helped lineages survive and diversify in California, and whether we see the same patterns in other native shrub lineages. And I should say just the broader picture of why this kind of research is important is that drought tolerance is important for the survival of all plants, but it's particularly pressing in areas like Southern California that are experiencing record droughts. So it's interesting to know how species adapted in the past, and if they were pre-adapted to drought, and if they might be able to do so in the future as the climate continues to change. So I'd like to thank everyone involved in this research and my funding sources, and NSF RCN from the Micromorph program, and Barnard and UCLA for support.
I might have missed this, but is there any difference in cell size, specifically maybe vessel element width, between these two groups that you studied?
Yeah, we looked at cell size, and I haven't yet looked at the vessels, but I do want to do that. But so far, it seems like, it depends on the cell type. I'd say like the cuticles tend to be thicker in the Cerastes species. But yeah, it seems like there's also, so I didn't talk about this, but there are like these large, what we're calling the hypodermis in the Cerastes species that they seem that the species don't have. But as far as other cells, I'm not sure that they're particularly different size. Yeah.
So if I remember Ackerley's paper, he claimed that the drought tolerance predated the Mediterranean climate in California. What's your opinion?
So there's kind of, I guess, two different ideas about that. So I guess the Ackerley and Axelrod have that opinion that the Mediterranean climate emerged around 5 million years ago. And there's kind of another group of Baldwin and Phil Rundle that say it might have actually been around 12 to 15 million years ago. So I think they have a paper coming out soon. But yeah, it will kind of affect how we interpret these results once I see that. All right, thank you.